Okay. Okay. All right. Okay, so at any point, please just stop me and just ask questions, okay? So unmute yourself and just ask the questions that you need to ask. So our first lecture is on platelet structure and function. You might already know some of this information if you took cell bio, you took a look at different cells, probably the platelets, but the platelets are a very, very important type of cell because they are the first type of cells that come to the site of injury. Whenever there is an injury in a blood vessel, okay, the platelets are kind of like the first soldiers that come to the site. Okay, so their function, even though they are anuclear, so they have no nucleus, they're really, really tiny. They have a huge function in the human body. So this chart shows you something called hematopoiesis. Hematopoiesis is the formation of your blood cells, okay? So it can be your red blood cells, your white cells, your platelets, okay? It can be your mast cell, okay? And we all know that all of these cells derive from your stem cells, right? And we know all the characteristics of the stem cells. I'm actually gonna do a whole lecture on hematopoiesis when we start hematology. So this is just gonna be a very brief review of what happens because I want you to understand where the platelets are coming from, okay? So we have the long-term self-renewing stem cell. Eventually, we may also have the short-term self-renewing stem cell that leads to the pluripotent hematopoietic stem cell. So pluripotent means that it has the ability to form or to divide into two different cell lineages. Okay, this stem cell right here can either turn into a common myeloid progenitor cell or a common lymphoid progenitor cell. So from the common myeloid cell, you get the white blood cells, okay? You get cells like the neutrophils, the monocytes, the eosinophils, and the basophils, also the mast cells and the macrophages, and from the common, and also the megakaryocyte. So the megakaryocyte is something that we're gonna talk about a little bit further in this lecture. Okay, you also get the erythrocytes, which are the red blood cells. Okay, so that all comes from the common myeloid. From the common lymphoid, what's going to derive from there are your lymphocytic cells, right? So your pre-B, pre-T, your B lymphoblast, which are the most immature form, your T lymphoblasts, your B cells, your T cells, and eventually your plasma cell, which is a derivative of the B cell. Okay, you're also going to have the natural killer cells. So this and the dendritic cells. So these are your B cells and these are your myeloid cells, okay? So megakaryocytes are considered to be myeloid cells. They come from the common myeloid progenitor cell. The whole process of cell formation, of blood cell formation is hematopoiesis, okay? So the megakaryopoiesis is the formation of the platelets, okay? So the platelets come from the big cells that are called megakaryocytes, okay? And these come from the common myeloid progenitor cell. A megakaryocyte is actually the largest cell in the bone marrow, okay? It has a very low nuclear to cytoplasmic ratio. It has abundant cytoplasm. The nucleus of it is multi-lobed, okay, so many lobes. It's very irregular, okay, and it has condensed chromatin. I'm going to explain all of that, and no nucleoli. Okay, the cytoplasm of these megakaryocytes can be bluish pink with abundant fine lavender granules. Okay, the picture is not the best picture, but this shows you the formation of a platelet, okay? So all of these blood cells, hematopoiesis, is happening in the bone marrow. It's not happening in the blood, in the actual blood. What you see in the actual blood are your mature cells, okay? You can see some immature or some growing cells or cells that are maturing in the blood only when there is some sort of deficiency. Okay, so that's what we're all going to learn about in coag and in hematology is all of the deficiencies that we see in the blood because the blood doesn't mature normally. Okay, or they get released from the bone marrow to the peripheral blood in an abnormal manner. So that's all the stuff that we're going to learn together. Okay, so you go from in the bone marrow having a megakaryoblast, which is the most immature form of a megakaryocyte, to a megakaryocyte, to a megakaryocyte that's actually forming platelets. And I'm going to explain the procedure and the platelets. Okay, so in the peripheral blood, what you see are the platelets. Okay, the cells that come to the rescue. That's how I look at them. They're like the ones that come to the rescue when there is bleeding, when there is injury. Okay, here's some definitions. Your megakaryocyte is the largest cell, and these are the cells that form the platelets. Megakaryocytopoiesis and megakaryopoiesis is the same thing. You can say it either way, okay? And it's the production of megakaryocytes, okay? Thrombopoiesis and thrombocytopoiesis, same term. You can use them the same way, and that is the formation of platelets. Thrombo, every time you think of thrombo, think of platelets, okay? And platelets are sometimes called thrombocytes, but the common name is platelets, so that's how we refer to them for the most part. Endomitosis, I don't know if you have ever read about this, I'm pretty sure you have. Endomitosis is the replication of chromosomes within the nucleus without actual cell division. Okay, it's also called endoreduplication. Okay, all right. So endomitosis is the replication. So the chromosomes are replicating, but there is no cell division. Typically, during mitosis, you have cell division and then you have the replication of chromosomes. But with megakaryocytes, that's not happening. That's why megakaryocytes are very special cells. All right, ploidy refers to the number of chromosomes that are present in the nucleus, right? So 2n diploidy, and anything that's greater than 2n is polyploidy. The nuclei, or not the nucleus, but the megakaryocytes are polyploidy because there's a lot of replication of the chromosomes that is occurring in a megakaryocyte. So this slide right here is the process of megakaryopoiesis and thrombopoiesis. Okay, thrombopoiesis is again, what is thrombopoiesis? Anybody?
The formation of platelets.
That's right. And megakaryopoiesis, the formation of a megakaryocyte. But you don't have to remember this chart right here by all means, please. I just want you to know that endomitosis is really what's happening here. Okay, megakaryocytes have multiple chromosome copies. Okay, they are recruited from that common myeloid progenitor cell, and they differentiate through several mutation stages. Endomitosis is just a unique mitosis. Okay, and it's very unique because it only happens in megakaryocytes. Okay, and that is the replication of chromosomes without the actual cell division. Okay, megakaryocytes arise under the influence of the transcription gene that's called the GATA1 gene, that GATA1 gene, plus the FOG-GATA2 genes. Okay, oops, sorry. And this is regulated by the cofactor FOG1. Okay, that's a cofactor. As the GATA1 and the FOG1 transcription slows down, the RUNX1 gene mediates the switch from mitosis to endomitosis. Okay, and under the influence of many other transcription factors, DNA replication proceeds to the production of 8N, 16N, and even 32N ploidy. Okay, So megakaryocytes use their multiple DNA copies, okay, to synthesize abundant cytoplasm, which eventually turns to be platelets. All right. So in order for a megakaryocyte to help release these platelets, it needs to have a lot of cytoplasm. And that's why there is endomitosis happening, because it allows for the replication of these chromosomes. You can get up to 32 N ploidy. So you know that it's a very high number of chromosomes that are dividing. Okay, so you don't need to remember these genes, okay? Just remember what's happening in the process of endomitosis and why that is so necessary in megakaryocytes. Now, megakaryocytes are recognized in different stages. Obviously, this is a cell that is coming from a stem cell, so it's going to differentiate eventually into a megakaryocyte. The cell is going to differentiate eventually into a megakaryocyte, and then eventually it's going to give us some platelets, okay, from the cytoplasm. So there's three different types. There's the MK megakaryocyte 1, which is the megakaryoblast. And this is smaller in size. The nucleus is round. There is about 2 to 3 nucleoli. Nucleoli are like the little eyes of the cell. They're little circles that lie within the nucleus. It has homogeneous chromatin. Homogeneous chromatin means that it looks very, how do you say it? It looks very, uh, uh, um, very soft, okay? Hard to tell what kind of cell that is, okay? It has high NC ratio, which means that the nucleus, okay? NC ratio stands for nuclear to cytoplasm, okay? The nucleus to cytoplasm ratio is high, which means that the nucleus is big in relation to the cytoplasm. That's what NC means, okay? So it has high NC ratio and it's polyploidy. That means that it has many multiple chromosomes present. The cytoplasm is typically dark blue. Another term for dark blue is basophilic, and it has many blunt projections. It can have many blunt projections. MK2 is the promegakaryocyte. It's a little bigger in size. The nucleus is very irregular or lobulated sometimes. You may or may not see nucleoli. It has condensed chromatin, okay? And the NC ratio is a little lower now because now the cytoplasm seems to be a little bigger and the nucleus gets a little smaller, all right? But it's still polyploidy. The cytoplasm is a little bluish, more like less blue than the MK1, and it's granular, okay? Okay, so the MK1 is actually the least differentiated stage of the megakaryocyte, okay? These cells right here, they cannot be reliably distinguished from other bone marrow cells. And and that's okay, and we're going to discuss things that are tests that we use that help us identify those cells for what they really are, okay? But when you have a very immature cell, even no matter from what stage it comes from, it's very hard to differentiate them in the bone marrow, so we have to do additional testing to really identify them, okay? MK2 is the promegakaryocyte, and this is a little bit more mature. So you can see the cell a little bit more. It's bigger. You can see like irregularities in the nucleus. You can see that the cytoplasm is a different color and it's granulated and all of that. The third stage is the actual megakaryocyte. This is much bigger in size. Again, this is the cell that is the largest cell in the bone marrow. It is multi-lobed. So the nucleus is multi-lobed. It can have 2 lobes or it can have 3 lobes. You can see in here, there's a little cleft, and then over here as well. It might be a little hard to see, but this nucleus here is certainly multi-lobed. It's irregular in shape. Nucleoli is not visible. It has very condensed chromatin. Chromatin is the structure that is within the nucleus, and it's what carries the DNA, right? And so it looks very condensed, very, very tight, okay? It has lower NC ratio, and it's polyploidy. The cytoplasm is pink and granular, but big, big cell. Now, do you guys see all the little platelets around it? You see the little tiny little platelets. So these are platelets, these little dots, those are platelets in our blood. All right. So megakaryocytes are in fact so big that you can recognize them. So if you're looking at a bone marrow, okay, you can see which ones are the megakaryocytes because they are so large. Okay, and this is just a chart from your book depicting the process of megakaryopoiesis. And let me just go back to this. In the process of megakaryopoiesis and hematopoiesis, there's a lot of proteins and regulators, interleukins that we need in order for this process to happen, okay? So during differentiation, cell division, replication, all of that wouldn't be happening if we didn't have all these regulator factors that help us along the way. And for example, as you can see here, in the formation of megakaryocytes from the common myeloid, right, you need to have TPO. Okay, TPO is a protein, a hormone that is needed for the formation of a megakaryocyte. Without it, we don't have that megakaryocyte, okay? Okay. And well, let me just go back to this. This is the differentiation from the stem cell to your MK1, MK2, and your MK3, which is actually the megakaryocyte. The megakaryocyte will create the platelets, okay, from things called the platelet processes, which extends out from their cell membrane, and then eventually these will shed into the platelets that you see in the blood. And I'm gonna get to that in a little bit. Okay, so here are some of the features of the terminal megakaryocyte differentiation stages. So you have MK1, MK2, and MK3, and the difference between them. It's just really helpful to know that your MK3 is the largest one in size, multi-lobe, no nucleoli. The chromatin is deeply and variably condensed. The NC ratio is decreased. So there's a lot more cytoplasm, no mitosis, no endomitosis. So there's something worth noting is that no mitosis. Mitosis is not happening in a megakaryocyte at all. But endomitosis is only happening during the MK and MK2 stages, okay? Once you get to the megakaryocyte stage, there's no more endomitosis happening, okay? The color of your cytoplasm, the granules that are present, and all of that is worth noting, okay? And here's another picture. This is actually a terminal megakaryocyte that is shedding platelets. So if you can see right here, this is your nice big megakaryocyte cell. You have your nice big nucleus that looks like it's multi-lobe and irregular, right, in shape. It looks like it's the chromatin inside the nucleus is very, very condensed. And here you see the pseudopod sort of processes happening, and then eventually these start to shed. So these are the platelets, okay? The platelets are fragments of the cytoplasm of a megakaryocyte. Okay, moving on. So as I said before, in order for megakaryopoiesis to happen, we need a lot of help, okay? We need lots of hormones and cytokines to help us. So TPO is one of the main hormones that need to be present and need to be intact in order for megakaryopoiesis to happen. TPO is thrombopoietin and erythropoietin is EPO, okay? We also need EPO for this to happen. The primary source for these two, thrombopoietin and erythropoietin, is from the liver, okay? Okay, so these hormones, they bind megakaryocyte and platelet membrane receptors. TPO plasma concentration is inversely proportional to platelets and the mass of megakaryocytes. It helps induce differentiation, proliferation, and maturation of megakaryocytes and thrombocytopoiesis and platelet release, okay? Again, without these hormones and proteins, all of this is not happening. And cytokines like interleukin-3, interleukin-6, interleukin-11, KIT ligand, granulocyte macrophage colony stimulating factor, GM-CSF, and granulocyte colony stimulating factor, GSF. These are also stimulating hormones, okay? So without them, it's not happening. So we need the help of GMSF and GSF to stimulate the differentiation. But these are naturally occurring interleukins and hormones that we already have in our body, unless there's disease and then you don't have them. Okay, we also have hormones and cytokines that control megakaryopoiesis. And what I mean by control is you can't be producing all the time. The body has the way it functions is that it knows when it has too much of something, and it uses certain proteins to be able to regulate that, or like to stop that process. So if it doesn't have those inhibitory proteins, then we have a big problem, right? So we need to have formation, and we need help for all of that, but also we need to be able to inhibit that as well. So the body makes proteins to help with that as well. So we have hormones and cytokines like platelet factor 4, beta thromboglobulin, neutrophil activating peptide 2, interleukin 8. These are all inhibitory hormones and interleukins that help us prevent megakaryopoiesis. So at some point, it helps us stop in certain checks. And then you have transcription factors like FOG1, GATA1, and FE2. These you don't have to worry about, but these are the transcription factors that take place in endomitosis, in the creation of megakaryocytes. Okay, so now let's discuss how we actually create these platelets, right? So now we know where our megakaryocytes are coming from, but now we need to know where our platelets are coming from. We know that it's from the cytoplasm of a megakaryocyte, but there's a whole process to them. So thrombopoiesis or thrombocytopoiesis is the process of the megakaryocyte forming a platelet or platelets. Each megakaryocyte can form about 2,000 to 4,000 platelets. So it's a lot, okay? But that's why in the blood, you see that our platelet numbers are huge, okay? Because they're really, really tiny structures and you need a lot of them in order for the whole function to be carried out. Megakaryocyte cytoplasm actually forms bulges or pseudopods that elongate into branching proplatelet processes. The platelets form from a bud at the end of these processes, and the DMS, which is the DMS is something equivalent to the plasma membrane of a megakaryocyte. The DMS provides the membrane for the processes and the platelets. So here it is. So the process of thrombopoiesis is caused by the breakdown of proplatelets. Okay, these proplatelets are actually the mature megakaryocyte membrane pseudopods, pseudopodial projections. These are the proplatelets, okay? So the reason why these platelets are being formed and released is because these proplatelets are breaking down, okay? And during the process, all of the membranes, organelles, granules, macromolecules in the cytoplasm, they're all being consumed during this process, okay? So thrombopoiesis really means just the formation and the budding of these platelets through the breakdown of these proplatelet processes. Is that clear? Or do you guys understand that? Okay. All right. So what's happening is that uh, what's happening is that as the cells mature, as our megakaryocytes come from stem cells in the bone marrow, right? So these cells, as they mature, they get closer and closer. They get moved closer and closer to the sinuses of our bone marrow so they can get closer and closer to the peripheral circulation, okay? So what you see is that these megakaryocytes, the big cells, try to extend these proplatelet processes across the endothelial cells into the peripheral circulation. Okay, the endothelial cells line the blood vessels and the bone marrow sinuses. So that's where the cell is so smart that it knows where it needs to go in order to drop off these platelets. Okay, if it doesn't have them ready, it's not going to go there. It's not going to go to where it needs to go. Okay. Okay, so shedding of the platelets into the sinuses is what the actual process is. It's that shedding process of these proplatelets, of this breakdown of the proplatelets is thrombopoiesis, is the formation of the platelets. So once these platelets get dropped off, they get released, it leaves behind a naked megakaryocyte nucleus. And so this nucleus is then consumed by the bone marrow macrophages. Okay. And then the whole process starts again and again and again and again. Okay. All right, so some unique features of megakaryopoiesis and thrombopoiesis. Again, megakaryocyte, it is, oh yes, so one thing to know is that as the megakaryocyte matures, the cell gets larger and larger and larger because the megakaryocyte is our biggest cell in the bone marrow. Now, typically with all of your other cells that you're going to learn about, or all of your white cells and your red cells, you start from a very large cell to a small cell in the peripheral circulation, okay? This is the opposite. So your megakaryocyte becomes larger as it matures. It is the largest cell in the bone marrow. It's polyploidy. Endomitosis in the nucleus is complete at MK. That should say MK2. It ends at MK2, okay? And you can have 32N up to 128N. The megakaryocytes form branching proplatelet processes, and then platelets form at the end of these processes and then are released into the bone marrow sinuses. And here it is, a nice, beautiful megakaryocyte with proplatelet processes, right? And so these get shed into the circulation. And again, platelets are the fragments of the cytoplasm. Once the platelets are released, it leaves this nuclei, this nucleus to be phagocytized by macrophages in the bone marrow.
Are the bone marrow sinuses basically just going right into the bloodstream then, like for the platelets?
Yes, yeah. So with the bone, so there's different compartments within the bone marrow. And as the cell mature, the cells get pushed through the bone marrow until they reach areas where it can seep into the blood, the peripheral circulation. The whole actual process of how this happened is it's actually not completely understood because there is a lot of interleukins, there's hormones, proteins, cell receptor signaling and things that happen for that whole process to happen. But the cells, their maturity points helps them move through the bone marrow into the peripheral circulation. And yes, and the sinuses are where they can cross through the peripheral circulation by the endothelial cells. So now platelets. Platelets are very, very special cells. They are very helpful in the body. They help maintain vascular integrity. they participate in something that's called primary hemostasis. It's the initial arrest of bleeding by forming the primary plug. Okay, well, we're going to talk about that in the next lecture. They help stabilize the primary plug through the participation in fibrin formation. They go through different stages when they participate in coagulation. They go through adhesion, secretion, aggregation stages, which I'm going to explain in the next lecture. They have a plasma membrane, which is a lipid bilayer. It has more than 50 receptors. Their phospholipids have asymmetrical distribution. During activation of these platelets, the polar phospholipids flip to the outer layer of the plasma. It has phosphatidylserine, which is PS, which serves as the binding site for activated coagulation complexes. They are non-nucleated cells, so platelets don't have a nucleus, and the reference interval in the human body in a normal adult is between 150,000 to 400,000. Now, when we talk about numbers in hematology and coag, we're always gonna, because obviously there's so many cells in the blood, right? So when we actually count them, we report them like this. We report them as 150 to 400 times 10 to the 9th per liter. That means you're just adding 9 zeros to this number, right? And that's per liter of blood, okay? So believe it or not, that's what we do in hematology, is we count your cells. We have instruments that help us count your cells, okay? So this is a reference interval, which means the normal amount that a regular adult should have of platelets. So if you were to look at the peripheral blood, you put 1 drop in a slide, okay? You let it dry, or you make a smear, you let it dry, and then you stain that, okay, with a special stain, and then you put it under the microscope. And you look at this and you say, okay, this is a normal adult. Okay, I see over here, there's red cells here. There's always going to be spaces in between red cells, depending on where you look. But if you're in a good area of the slide, this is what you should be seeing. These are your white cells. These are your typical neutrophils, which is one of the white cells that we have in large numbers. And here are your platelets. So the platelets look like dots, basically. That's what they look like, okay? This is in the peripheral blood, okay? And looking at the platelet a little bit closer and looking at their phospholipid membrane, it's gonna be very important because platelets have receptors in them that we need very much in order for coagulation to happen. So if they didn't have the receptors, it wouldn't be able to participate in the primary plug and then in the secondary platelet plug. And so nothing would fall into place. So the membranes have the, obviously, you know what the membranes look like. Typically, you have the fatty acid tails with the polar head groups. You have the esterified cholesterol. You have carbohydrate side chains that extend into the plasma to form certain receptors. And this is typically an example of one of the receptors. Remember, platelets have more than 50 receptors. We don't have to learn all of them, but we are going to have to learn the names of some important receptors in the platelets. Okay, it has the transmembrane glycoproteins and the microfilaments in the cytoplasms. So I'm just going to repeat everything that I just said. Normal platelets are very, very tiny. Remember the size of them, okay? So if you compare this cell to the red cell, or even to the, even if you compare it to the white blood cell, the neutrophil, it's very, very tiny. They look like dots, okay? So the platelets are about 2.5 micrometers in size. And they have a mean platelet volume of 8 to 10 femtoliters. Femtoliters is a way of identifying the volume of something that's really, really tiny. Okay, so femtoliters. We also have something called stress platelets, okay? Stress platelets or reticulated platelets can appear in the circulation, and that happens when the body is compensating for thrombocytopenia. Thrombocytopenia is a decrease in platelet number. Thrombocytopenia, okay? These platelets are larger in size. They carry free ribosomes and fragments of the rough endoplastic reticulum, and they are detectable using flow cytometry, which is a test that we use with nucleic acid dyes. So they're just bigger platelets. They're called stress platelets. They're bigger than 6 micrometers in size, and the volume is a little bit bigger as well. They're cylindrical. They look like these red cells that we call reticulocytes. I'll explain that later, but they have ribosomes. They have free ribosomes and free ribosomal endoplasmic reticulum fragments in them. And I'll show you the picture of a stress platelet. So these could be big stress platelets. So they look like they have fragmentation inside of them. Now, these can be, again, it's a response to stress, to something that's happening, and you can start seeing them when the body's trying to compensate for a low platelet count, a low number of platelets. Okay, platelets also have, remember, they don't have a nucleus, but they have a lot of organelles and structures within them that have very important functions. So platelets have dense bodies. There's also these dense bodies, they have about 2 to 7 per platelet. They're like granules, sort of, and they contain ADP, ATP, serotonin. They contain calcium and magnesium, okay? Very, very important to have calcium in them, and I'm gonna explain why. They have alpha granules, more alpha granules than dense bodies, about 50 to 80 per platelet. Okay, these granules have fibrinogen. This is called factor 5, okay? This is called von Willebrand's factor, plasminogen, inhibitors, growth factors, and other proteins. You don't have to know what that is right now. I'm gonna explain all of that in the next lecture, but it's good to know that alpha granules are within our platelets. They have very, very important components that we're gonna need for coagulation, okay? They have the glycocalyx that's on the surface of the outer plasma membrane. On the surface, on the top, it contains mucopolysaccharides, glycolipids, albumin, fibrinogen, and other plasma proteins, and they give the platelets a negative charge, okay? So this whole glycocalyx situation with the platelet membrane is very important because if the platelets don't have a negative charge, they will flow through our blood, but then they'll bump into each other and they'll stick together. Okay. Now, because they have this negative charge, they tend to repel each other. So that way, when they flow nicely as an individual platelet, unless they are needed when there's a site of injury. Okay. When there's injury, they're needed, and then they get aggregated. But otherwise, for them to flow, they need to have a nice negative, kind of like a negative force, like a negative protection around them so that they don't all aggregate together. Does that make sense? And that's something that the glycocalyx provides. All right. Other organelles are the surface connected canicular system. And this is a canal, extensive canal system in the cytoplasm that connects to the outer plasma membrane. And these alpha granule contents are secreted out of the cell, and molecules are brought into the cell by the surface connected canicular system, okay, this SCCS. So it's kind of like a canal within the platelets, okay? So it connects the outer plasma membrane to the inner side of them. And then you have the dense tubular system. It is the activation center for the platelets. It's also a storage reservoir for calcium, and it has enzymes that are needed for platelet activation. Okay, it does not connect to the platelet surface. Remember, these organelles are so important in a platelet because we don't have a nucleus. So normally a cell that has a nucleus will be able to do a lot of stuff, right? the mitochondria, the DNA, the chromosomes, the this, the that, but the platelets don't have that. It's a whole nother type of cell. But these organelles are so individually important that they all have a function in the overall function of the platelet, if I'm making myself understood. All right, and that is a picture, a diagram of the platelet membrane, and also all the receptors and everything that is associated with the activation process of platelets. We don't have to memorize any of this. This is just a nice image that helps us understand what is happening in a platelet. Don't get overwhelmed by this picture though, because there's a lot in here. But let me just point a few things out, okay? All right, so this is the plasma membrane in the glycocalyx, right, all around it. There is the surface connected canicular system, right? You have the let's see, the alpha granules, the dense granules right here, as you can see. You have the cytoplasmic microfilaments, collagen. You have certain receptors. If you can see here, there's one receptor called glycoprotein 1B95, glycoprotein 6. It's a very important receptor as well. Okay, we have the DTS right here. So again, not to memorize any of this, but just to have an idea of where these organelles are, where they lie, basically. Okay, so the function of the platelets, well, I'm going to talk about this now, and I'll talk about it again in the next lecture because we get into more detail. So the platelets come from the megakaryocytes. We know that they have a very important function. Now, their function is broken down in several steps, 3 main steps to be exact, okay? The first thing the platelets do is they adhere to the site of injury, okay? So let's say there's a break in a blood vessel. The body is automatically going to know what to do when there's a break, okay? And the platelets are these very smart cells that they know something is happening, so they get called to that site of injury. So in the first step is going to be that they are going to adhere to that site, okay? So in conditions of rapid blood flow and high shear stress, that's typically the type of blood flow that you find in arterioles and capillaries, okay? You're going to need something called von Willebrand's factor. Von Willebrand's factor is a protein that we have in our body. It circulates in the body, but it's needed because the platelets can't just go bind when there's high stress in the blood flow because they kind of get washed away. So when there's high stress in these capillaries and arterioles, the platelets are flowing through, but then they find this von Willebrand factor protein and they bind, okay? So in high blood pressure, high shear stress in blood flow, in arterioles and capillaries, von Willebrand's factor binds to collagen in the exposed subendothelium, and then it unrolls. Now, what happens is that von Willebrand's factor is a protein that is circulating it kind of like a little ball, okay? It looks like a ball. It circulates through the system. But then when it's called to the site of action, it goes to where it needs to go, and then it opens up, okay? So when it opens up, when it unrolls, it exposes its receptors, okay? So von Willebrand factor comes, it binds to collagen in the subendothelium, and it opens, okay? Now, the platelets are rolling through, right? The platelets are rolling through. So then the platelets find this von Willebrand factor, and they bind to von Willebrand factors through their platelet receptor called glycoprotein 1B95, okay? Now, pretty much everything that we do in coag is given to you in Roman numerals. I don't know why, honestly, but that's what the name is of this platelet receptor. It's 1B95. So that's the first platelet receptor that is pretty much needed in order for the platelets to adhere to the site of injury. Okay, now van Willebrand's factor is there now, right? So it kind of forms a bridge between that receptor and the collagen to the subendothelium to initially adhere the platelets to that subendothelium. Now, this is only when it's in arterioles and capillaries because the platelets get washed away. So think of just the platelets not being able to adhere because there's just too much blood flow in those areas. Okay, now if the platelets can bind directly to the site of injury in other places that is not such high stress of the blood flow, so they can bind directly to the collagen by their glycoprotein receptor 6. Okay, and that generates a signal for platelet activation. So platelets bind and then they start to activate. Okay, that's called the outside in signal. Okay, when the platelets activate, they undergo a shape change. So normally they're nice and round, but now they they turn into a different shape, kind of like a spiky like shape to them. And then they expose another receptor called the glycoprotein 2b3a receptor. Okay, that becomes activated on the surface of the platelet. Okay, so in a resting platelet, just a little side note, okay, the two chains are separate and inactive. Okay, in the activated platelet, there's an inside out signal that causes the two chains from a heterodimer, which then activates the receptor so it can bind to different types of ligands. Okay, so this is just what this means is just there is a shape change. Okay, and because of this activation, this shape change, you get a different receptor that gets exposed, and 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 this is coming from an inside out signal. Okay, so that means that their phospholipids in the membranes have flipped, and now you have a different kind of surface that's showing in the surface of the platelets. We have this in more detail in the next lecture. This is just an overview. Okay, once the platelets are activated, then there is something called platelet secretion. Platelet secretion happens because they release certain um, um, secretions. Okay, activated platelets synthesize and release thromboxane A2, TXA2, and the content of their granules. Remember, we talked about platelets having these organelles, right? Dense granules, alpha granules, and these dense granules, alpha granules, they have things in them, right, that help in the function of the platelet. So here is where all of that is getting released. Okay, so platelets start secreting substances, all right, so thromboxane A2, and then the contents of the platelet granules. Okay, so there are two main secretory products here. The substances get released from platelet granules, and that requires energy. So ADP, serotonin, von Willebrand factor is also within platelet granules. And then coagulation factor 5, which participates in the formation of fibrin on the surface of activated platelets. Okay, and during this whole process, then you have the formation of thromboxane A2. So remember these organelles, although the platelet doesn't have a nucleus, they have these organelles that are very, very, we need every single one of these substances for more platelets to come, okay? Because we need to arrest that bleeding. So with just one or two platelets, we're not going to arrest any type of bleeding, okay? So we need adhesion. We need the platelets to activate. We need them to secrete substances, okay? And we need them to aggregate, okay? The platelets eventually start aggregating. And what happens is fibrinogen, which is another protein, forms bridges between glycoprotein 2b3a of adjacent platelets to form what we call the primary plug, okay? But Willebrand factors also binds to glycoprotein 2b3a molecules on adjacent platelets. So now we're starting to aggregate. So we're forming that primary plug. We need a plug. We need to first arrest this, okay? And this is just a picture of what a resting platelet looks like and what an activated platelet looks like and how it differs in change and the receptors that it that it shows. So the reason why we have something called resting platelets is because this actually prevents accidental aggregating and accidental clotting, because your platelets play such a big part in clotting. If they are allowed to bind all the time, then we would be having clotting issues, right? So normally what it does is you're in a resting stage, okay? So there's no activation, there's nothing happening. They're discoid, they're bond-concave, there's no phosphatidylserine, no p-selecting. The glycoproteins receptors are inactive, okay? They're there, but they're inactive. With your activated platelets are a little bit different, because your activated platelets are actually not discoid in shape, but they are flat. They have pseudopods, okay? They have phosphatidylserine. They have p-selectin, which you can see in here as CD62 on the surface. They have active glycoprotein 2B3A. A coplatelet is a platelet that has been activated by collagen and thrombin. So a resting platelet is here, okay, and this is a coplatelet. It's a coplatelet that has been activated. So the receptors are there, but they are active, okay? In the resting platelet, the receptors are there as well, but they are inactive, okay? So remember I talked about that change in shape due to activation. So a resting platelet will be kind of discoid in shape, biconcave, and the activated or the coplatelet starts to change shape. So it will not be discoid or biconcave, but maybe flat, and it has pseudopods, so kind of like spiky looking. That picture doesn't really do it justice, but there's other pictures that I will be showing you where you can see that difference. And this is just a chart from your book that shows the major platelet receptors. And the major ones are your glycoprotein 1A, 2A, okay? And it shows you your ligands in here. Glycoprotein 6, that's a very important one. The ligand is collagen. Glycoprotein 1B95 is another big one. And the ligands are von Willebrand's factor, thrombin, which is another protein I'll talk about, and glycoprotein 2B3A. It's another one that's important. Now, do you guys see something here called cluster designation? Cluster designation is, they're called CD markers, and they're found on each and every single one of our cells. CD markers are the markers that we can identify through a test called flow cytometry, and it helps us determine the stage and the lineage of the particular cell. Now, these receptors are also designated as CD markers, okay? Also designated as CD markers. So typically, when we want to count platelets, when we want to identify platelets, we can use flow cytometry as well. And we pretty much look at the most common ones, which are your CD41 and CD61, which is the platelet receptors 1B, platelet receptors glycoprotein 2B3A, okay? And also CD42 is another common glycoprotein that we identify in flow cytometry. Okay, so I'm going to stop here. I know this was a lot, but again, so this is chapter 4, but the bulk of this lecture you can find in chapter 10. Chapter 10 gives you a really nice explanation. I have the book with me right here, of platelet production, structure, and function. So when you have a chance, take a look at that, okay? All right, I'm gonna stop this recording. Oh no, hold on. Pause recording, stop recording, okay.